lot, Dr. Harikrishnan, Dr. Kartha, Director Shri Chitra, for inviting me for this very important meeting. I think this is a dream which is coming true for ICMR also. The heart failure program is moving fast because of all of you. So I was asked to present something on biobank because heart failure biobank is one activity which has been undertaken under our Center for Advanced Research and Excellence at Shri Chitra. So coming to the history of biobank, the first time this term came into the scientific journals, it was in 1996 when Loft and Paulson, they wrote about this term in their article on cancer research, cancer risk. But later on, with the era of omics coming into being, the IT and the big data and the large electronic databases, came the idea of personalized medicine. Now, when we talk about personalized medicine, we do understand that if we want to do research in that direction, we'll be needing large collection of patient samples, which are well annotated. And then came the requirement of a biobank. Now, as far as biorepositories and biobanks are concerned, these terms are generally used interchangeably. Just give me 1 minute. These terms are used interchangeably, but initially we used to think that biorepositories are the ones which are somewhat uncontroversial, being situated in the seclusion of pathology institutes or in the laboratories. The investigators, they were the persons, nodal persons for these biorepositories. But with the biobanks, there came a potential to use the existing collections besides opening new biobanks. The scale is different for biorepositories and biobanks. Biobanks are much more larger in size and they cover a large range of diseases. And there are a number of institutes which can be involved both within the country and globally. Now, this is one slide which I tried to copy from a very recent journal, The Role of Modern Biobanks. If you look at it, then since their inception in early 2000s till date, we see the role is expanding. With the advent of biobanks, we have a chance of doing large number of multicentric, multidisciplinary studies, including genetic studies, environmental or lifestyle risk factor studies. And the most important thing is the samples which are lying with us, along with the clinical data or epidemiological data, they can be utilized for undertaking studies using novel techniques 20 years later also. So that is a promise which the biobanks bring with them. And as a result, it's a multidisciplinary approach, different kind of people, all from clinicians to researchers to bioinformatic people. They are all involved in that. And what we suggest is that when you are harvesting the sample, then at that early age stage only, you should involve the right persons. For example, if you are going to store tissues, then a pathologist is required at a very early stage so that you are not storing the blood clots or the necrotic tissues, which cannot be used later on and drain your resources. Similarly, for sample transportation, storage, retrieval, you require technical know-how, and there are people who are important. There are people with that kind of know-how, and everyone has to be involved in. So there are different types of biobanks. We all know that there are population-based biobanks, disease biobanks, case-control biobanks, tissue biobanks, and biobanks with clinical trials. Now, just for your information, ICMR has also started a stroke clinical trial network, and we are hoping that for heart failure also, we'll be having similar kind of clinical trial network. And once this network is there, there is always a hope of building up a biobank also, which can be utilized for research. In case of heart failure biobank, which we are establishing at Shri Chitra, this is a disease biobank. There are so many international biobanks, and it will be difficult to list down these in one slide. So some of them I chose randomly. UK Biobank is a very big biobank with 500,000 samples. Then Japan Biobank, European Biobank, Chernobyl Biobank, and so on. There are disease repositories also. Then there are biobanks in India also, which are being used for NCT research. One of the very important one is that at NIMHANS, the Brain Biobank. With again, for your information, what we are trying currently to do is this biobank was initially supported by ICMR, DBT, and DST. We have been supporting this through CARE for the last 5 years, but now we want that the Brain Bank at NIMHANS be expanded to multiple centers in the country. And the importance, this is very important at this stage because the number of autopsies in the country have declined. So very few centers are there. We were able to identify only 3 more centers, PGI, KEM, and AIMS Bhubaneswar, which are ready to do autopsies of that size so that the tissue can be included in the brain bank. That's an important activity which we are hoping to undertake. Then there are advantages of a biobank. The biobank have large sample size, and the cost, you will say that the cost is very high for establishing a biobank. But if we look at the cost reductions which happen because of sharing of the samples, because of making a large volume of samples available to the researchers, I think the costs are quite low in that sense. Then a single ethical approval can be taken which will lessen the time for individual biobanks. There is an infrastructure requirement for the biobank. The blood samples are the samples which are normally collected in a biobank. So I thought of giving the pro and cons of having these kinds of samples. We can have dry samples. Yes, they are easy to collect, small storage spaces they require. But the problem is they are sensitive for fungal attack, and they require adequate drying also. Then whole blood, which can be stored at -20, and serum samples, which require to be stored at -80 degrees centigrade. So these require large storage spaces, but they are very easy to collect and they remain viable for decades to come. Now, for the tissue samples, we require these to be stored in liquid nitrogen at -196 degrees centigrade, but the cells remain viable for a long time. And these can be, you are actually with an unlimited supply of cells for a very, very long time to come. But obviously, these are very difficult to obtain. You require a different type of specialization to collect them. You will require pathologists to be a part of your team, and you will require liquid nitrogen also. If the biobank is very large one, then in that case, it's better to have nitrogen plants. The other requirements of the biobank are that we require a consistent database to meet standards of international compatibility. These have to be real time, they have to be easy to assess and user friendly. Security is an issue with them because we require data protection. The protocols have to be standardized. So this all comes with information technology. The IT can provide you that kind of support. So biobanks are something where all kinds of sciences have to come together. It is a teamwork which is very important, and very few people want to take that kind of responsibility. So that ownership is very important in this case. And bank, as you know, bank has to give the things to the others also. The loans have to be dispersed, and similarly here the samples have to be given to the researchers. Only then it becomes a bank. Then the laboratory facilities you will require, it's not that only the larger labs have to participate in this biobanking facility. Even the smaller ones, they are required for sample preparation. The larger ones, they can do the advanced analysis. But everyone is there in a team. So for data protection, we require anonymized samples so that we are not able to track the identity of the donor. But on the other hand, the biobanks may also require to go back to the donor when the data, when the analysis requires us to do so. So those kind of chains have to be built up, encryption and de-encryption. Similarly, coding and decoding. So these kind of security checks have to be there. A standard pre-analytical coding for biospecimen spec. This is the tool which is used for anonymizing the samples. This is recommended for maintaining a predictable and restorable sample collection. There are ethical considerations for a biobank. The donor, this is the most important thing and most difficult thing. The donor has to be informed. The donor has to understand the importance, the significance of these biobanks. If you all may be knowing that, for example, in America, this is becoming difficult. The people, they are not trusting that much kind of thing. So you have to build a trust relationship between the biobank and the public too. Public is an important player in a biobank, and their trust is very important. Can I have 1 minute? Can I have 1 minute?
Yeah, please go ahead. Yeah.
Yes, please.
Right to withdraw is an important thing. I will not go into the details, but there are different ways in which the participant can withdraw from the biobank. Now I come to, there are guidelines for concerning the biobanking issues. ICMR has also got its ethical guidelines, but the details about how to bring up a biobank, they are provided in different guidelines which have come up in the recent times. So, but why do we need a heart failure biobank? We have a consortium of heart failure specialists from different parts of the country in our heart failure registry. So, and we do know that we face a challenge of our population being a unique one with diverse culture, and we need to study our population to understand the heart failure in our population. So this is the heart failure registry. You know that everyone knows that there are 53 running centers in the country, and we have collected data of around 10,000 patients from 53 centers, and they are being followed up for a period of 1 year. We plan to follow up this cohort for a longer period of time. Once the study is over, the cohort will be supported by ICMR for a longer period of time. So this is how it goes. The biobank will have the specimens with it as well as the data. The data will include personal identifiers, demographics, clinical, and the follow-up data. And we are hoping that when we analyze, it will be resulting into not only publication but translation products too. And in the stage 1, what we are doing is we are building up a biobank at Shri Chitra so that all the procedures are standardized. And in the next stage, we'll be moving to other centers in the country, and we'll see that whether we will go for regional or for central biobank. So different kinds of biological materials are being stored at the biobank, and we are using, this is how we are coding it. As I said, that there is a SPREC V1.0 code, which we are using for coding the samples. And it does look that why we are taking so much of pain to set up a biobank, but the fruits are going to be good for this. If you look at the UK Biobank, this is changing the way we are doing science. This is changing our way of we are dealing with the public health problems, as you can see from the COVID-19 publication, which came just recently on February 3rd, 2021, showing that the antibodies, they can remain for 6 months. You can use these facilities for the raking public health problems. With this, I will say that each morning we are born again, but what we do today is what matters most. Thank you so much.
Thank you, Dr. Meenakshi. I think we'll now move on to the recap. Dr. Hisham and Dr. Venugopal. Any questions for Dr. Meenakshi before that?